Hi, I'm Dr. Elisa Domeno, and this time we're going to talk about the erythrocytes, or the red blood cells. Let's talk about where you get it. Most of the time, when collecting whole blood, you'll need to collect it from the veins. We call this procedure venipuncture. So primarily what's done, especially in adults, is the use of the cubital vein on the arm, right at the seat of the elbow. The veins are more superficial, they're thinner, and the best part is they have lower blood pressure than arteries. So when you need to conduct venipuncture, there's less likelihood that anything's going to spray out and you'll have a nice, even, predictable draw. Also, however, when you need to collect from peripheral sites, we use peripheral capillaries. Sometimes these are sites such as the fingertip or even earlobe. And once in a while, some individuals need to collect arterial blood. Now, this is usually for very specific procedures, such as to look at the efficiency of gas exchange of the lungs or overall respiratory performance. Red blood cells can account for nearly, 99.9% of all of the formed elements of blood. As we saw earlier in our previous videos, the hematocrit is quite a high percentage of just red blood cells. And they're red because they contain a certain pigment molecule, which has a secondary transport function. It's known as hemoglobin. Now that function is to transport oxygen and carbon dioxide around the body. So how do we measure red blood cell abundance? Basically, it's actually in a very small amount. The two that we use primarily, one is for histology. So this is where you would take whole blood and you'd put just a drop, in this case a few microliters, and you could do a cell count. And the reason we use the microliter, which is a very, very, very small volume, is if you look at the abundance of red blood cells in, say, men and women averages, they're in the millions, actually, per microliter. So it's quite a lot, quite a few of these teeny tiny little cells. Also, another way that we can measure it is using the hematocrit, which we saw in an earlier video. So what this does is it takes a vial from a blood draw and it spins it using a centrifuge. The heavier elements, such as red blood cells, tend to fall to the bottom. The medium weight ones, such as white blood cells, form a whitish pinkish band in the center, and then a yellow band is left on top, and that's the matrix or blood plasma, the lightest weight component of blood. So we also talk about this hematocrit after it's been spun down as being the VPRC, or the volume of packed red blood cells, because obviously they're going to be packed down by the centrifuge. So generally the hematocrit can be different for men and women, and also it depends on what's going on in their lives or their health at that particular moment. So it's also important to remember that this changes and these are averages. Hematocrit for men is typically around 46% red blood cells, as well as 42% of red blood cells found for women. So the structure of red blood cells is a little unique. As we know, they look more like donuts than they do the conventional cells, and that's due to a few things. So first off, let's talk about that structure as to why you would have it. It's specialized to transport oxygen and carbon dioxide. Quite frankly, it means that because we don't have a nucleus and we don't have that big beach ball shape, now as much surface area as possible of the cell membrane is in contact with things like the hemoglobin that's stored inside of the red blood cell. It means that the maximum amount of exchange between oxygen and carbon dioxide can be made. So it's actually really great. We don't have all of that useless space in the center of the cell that can't pick up or transfer any of these dissolved gases. Anyway, so it increases the rate of diffusion between cytoplasm and plasma, primarily with the dissolved gases, but also with things like nutrients, electrolytes, and wastes, because it now has a ton of surface area that can just directly contact. What else? Oh, of course, that's right. It's flexibility. Believe it or not, if you could reach out and grab a red blood cell and squeeze its sides together, it would squeeze just perfectly fine and flex right back into its original shape. And that's very helpful when we need to squeeze through these narrow capillaries at the most distal regions of the body. Other things to know, and we'll look at this in a bit, is the fact that during red blood cell formation, the way that they get biconcaved or donut looking is because they lose a lot of their organelles, especially the nucleus. The whole nucleus actually gets chucked right out. Because they don't have a nucleus, we don't undergo cell division. Let's look at formation. So originally, during embryogenesis, most of the red blood cells are formed in the yolk sac, actually, and they move eventually to places like the liver and the spleen, which will have subsequent roles in adult hemus, blood health, and also these little teeny tiny centers near the ossification centers called seed bone marrow as a fetus. As an individual develops, it's pretty much isolated to the bone marrow, the red bone marrow, really. Red bone marrow tends to concentrate from the site of all of the bones in the body during development as a fetus, very young children, eventually to the long bones. Hematopoiesis means this. It means red blood cell formation. Well, actually, it means blood cell formation is what it means. Red blood cell formation is erythropoiesis. So hemato for blood, poiesis, mitotically active stem cells, making new cells. And then leukopoiesis is what we refer to as a cell moving from a stem cell to a white blood cell. So the cells or the stem cells that we were referring to are what we call hemocytoblasts. So let's dissect that word for a second. Hemo meaning blood, cyto meaning cell, and blast referring to mitotic or stem cell-like activity. So it's a stem cell-like cell that is undergoing mitosis, making new cells, and it works specifically for the blood. Let's talk about the anatomy of the red blood cell. So the red blood cell itself, here as you can see, the biconcave shape. This is a blood smear over here in your far left side of the screen. And as you can see, they're teeny tiny little donuts, little pink frosting almost. Very hard to find for most students the first time they try to find it under the microscope. After a little bit more of a close-up scanning electron microscopy image, you can see that if we give a transection to the cell, we flattened out, there's no nucleus in the middle, and that's what gives us that donut shape, and they become relatively thin and flexible. They have a lot of cell skin or cell membrane to move around. So red blood cell formation continued. So this is also referred to as erythropoiesis. We know that the yolk sac is the primary site of formation for embryogenesis, but as we mature into adults, we stay in the red bone marrow, particularly of the long bones. As you can see up here, this is a cross-section of one of those bones. There's two kinds of marrow in there, yellow marrow and spongy red marrow. Now, hematopoiesis occurs in the spongy red marrow, not the yellow marrow. The yellow marrow is primarily adipose tissue or very, very dense, very rich fats. So this red bone marrow is also referred to as the myeloid tissue. So the formation of the red blood cell requires quite a few things. It's one of the reasons that vitamin B12 and vitamin B12 deficiency related anemias such as pernicious anemia can result in low volumes of red blood cells. So if you take a look at this little chart, we have a stem cell over here on your left-hand side, hemocytoblast, and it begins to commit. It's going to become a red blood cell. So if you look at the bottom of that second darker green panel, it says pro-erythroblast. Pro meaning now it's decided. It's proto-erythroblast. And once it commits, it begins the developmental pathway. Phase 1 is ribosome synthesis, or basically they make a ton of hemoglobin, the protein that seats at the center of it, a little tiny ion of iron. That iron ion is attractive to both carbon dioxide, but definitely oxygen. So we make a ton of this protein seated with a ton of this iron, and we do so much that we actually push and constrict the nucleus. So as you can see during phase 2, at the beginning of the normoblast, You can imagine that inside of that kind of tannish, pinkish interior is just chock full of hemoglobin. Eventually, it's made enough hemoglobin and it does something completely unexpected. It chucks out its own nucleus. Yes, folks, it ejects it, kicks it right out of the middle of the cell. It moves forward into its reticulocyte phase. So the reticulocyte phase is the compaction of proteins and also eventually plop, the sinking of the center component, making a erythrocyte, all nice and red and donut looking. Let's talk about hemoglobin for a minute. So hemoglobin accounts for like the lion's share of what's inside of a red blood cell. Because at this point, it's not making new cells. It's not going to be undergoing mitosis. It's just transferring stuff. And the most important thing that it can transfer at any given time is oxygen and carbon dioxide. So what it is a globular set of proteins. Globular meaning that there's two separate proteins and they just stick together in a big glob, a protein glob, and that's why we call them globular. Science naming, folks. It's so impressive. Anyways, inside of these globular proteins are little tiny parts we call subunits that contain a heme, right? Hematopoiesis, hematology, it all starts here. And that tiny little protein sits an iron atom. And remember what iron does. Iron attracts oxygen. If you think of any kind of rusty iron nail or rusty iron, I don't know, rooftop or something. I don't know too many things that are made out of iron, I guess. But anyway, they rust. Iron rusts. It turns that red, rusty color. And what that is oxygen working on it to decompose and separate the components of that giant iron sheet, for example. So iron is really, really attractive to oxygen. And so we've seeded quite a bit of oxygen, or quite a bit of iron inside of these hemoglobins, so we can attract basically and take in lots of oxygen and lots of carbon dioxide. Works pretty well. Side note, other animals have used copper, which is really interesting. I guess there's some oceanic types. Right, so as long as it attracts, I guess, oxygen, you're good. But we seem to have the best luck with iron. All right, so we're going to use it to attract up our oxygen. However, so whereas the oxygen binds specifically to the heme subunit, carbon dioxide wobbles a bit and doesn't bind as directly to the iron heme unit. It tends to bind more to the globulin subunits. So things to know about the globulin subunits is they're folded with a specific set of charges on one side as opposed to the other. This is what assists in the acquisition of carbon dioxide. So here's the picture, at least of the heme that I like. So at the top, you'll see a giant red blood cell. And if you move a little bit to your left, you'll see a purple hemoglobin, right? So it looks like there's all these proteins, and there's something in the center of them, something kind of silvery with a red dot. Well, let's blow that up too. Go a little bit more to your left, and you'll see a true heme. So this is a heme group, separate and alone, and in the center is that iron, so attractive to oxygen. So far over in the black box, gosh, I really wish my pointer and marker were working. I'm so sorry, guys. You'll see up here an oxygen or an O2 atom. See, O2 is going to be the red that's sticking on its two little oxygens bound together. And they're attracted to and bound to this iron that's in the center, right? So that's how it attracts and binds for a heme group. Down below is another representation of basically hemoglobin. So as you can see, it's 1, 2, 3, 4 all together, and two alpha subunits and two beta subunits. And all together we have 4 little irons trapped in there. That means we can transport 4 oxygen molecules, right? O2s for each hemoglobin we have. Very pretty, really organized, huh? So what happens when we destroy red blood cells? Because they actually wear out their welcome after a while. Just like a fence rusts or a nail rusts after too much contact with oxygen, so does, if you look down in the bottom of your screen, the red blood cell rust out to a purplish methyloglobin cell. Methyloglobin molecule, causing the red blood cell now to look a little more purple. It means that we've now gone from iron with a 2 plus to an iron with a 3 plus. So we've actually changed the electron capacity and made it less effective for holding on to and picking up oxygen. So things that happen also inside of each one of those red blood cells as it begins to convert hemoglobin to methylhemoglobin is enzyme activity is going to drop. Membrane lipids are not going to get really remain as stable, and we start to have less retention in overall cell structure, right? So it kind of starts being less flexible. So these aging cells are culled out by the liver, but especially the red blood cell graveyard, also known as the spleen. Globin itself is not wasted. We digest it. Yep, you digest it down into amino acids, and of course we try to recycle as much of the iron as we can. So not every single one of the irons in the heme groups have gone to a +3. So we'll still be able to reuse the ones with proper charge. Peripherin is now being reduced to bilirubin, causing that yellowish look as basically as it's being shattered open. So it's one of the byproducts. So whenever you destroy red blood cells and you smash them open and you start processing peripherin into bilirubin, that presence of bilirubin, which is a yellow compound, starts making everything look a little more yellow. So individuals who have jaundice, for example, down here, it's very common apparently in Albuquerque, New Mexico, for neonatal jaundice. So it's something to really look out for. Yellow skin, yellowing of the eyes, and you'll also see some other things such as kind of a darker urine. Sometimes they get sick, but definitely very sleepy and they don't want to feed. That yellow skin, yellow eyes are a dead giveaway. So jaundice would start appearing in those individuals. Also, jaundice is a symptom that shows up in many other types of red blood cell irregularities, which we'll look at a little bit later on. It's anemia. So anemia is one of the pathologies that's basically just an overarching term that describes any of the reduction in the blood's ability to not carry as much oxygen as it needs to or is expected to. It can be caused by a lot of things. Not enough red blood cells, maybe the hemoglobin has some problems, maybe there's a lower blood volume, right? And some of the symptoms of anemia are things like exhaustion, tiredness, muscle fatigue, weakness, cloudy headedness, just not being able to think straight, generally a lack of energy. So anemia can come in a couple of different types, right? So we talked about insufficient red blood cells, but let's also talk about low blood volume. So hemorrhagic anemia, meaning someone who's just lost a lot of blood. It can be sudden, it can be over time, it can be chronic, happen a lot, and it can be due to many different parts that are unseen. So it can be, say, an external laceration or an internal one, say somewhere in the intestines or digestive tract, anywhere in the body really. Hemolytic anemia is another one where there's too many red blood cells getting destroyed or rupturing. This is likely to occur with things like mismatched transfusions, certain types of drugs, and of course infections. Aplastic anemia is a little different. So this means that most of the components of blood are working. However, since there's been destruction of the red of the red bone marrow, which is the birthplace of all of our RBCs, we're not able to regenerate as quickly enough red blood cells as the body needs. Other types of anemia include things like having simply a low hemoglobin count per individual cell. Some of them are simply because there's not enough iron being absorbed into the diet, and so even though enough red blood cells are present and everything else seems great, we're just missing that one crucial nutrient in our diet. Megablastic anemia, also known as pernicious anemia, results as B12 or folic acid insufficiencies. So that folate, folic acid, super important stuff. Also important for preventing neural tube defects in the very earliest stages of fetal development. So just put that one in your pocket. Okay. Hemoglobinopathies are several different categories which can range from mutations to mutagens being present in the individual's environment, which lead to hemoglobin being irregularly produced and also then subsequently not able to properly carry either iron itself or transfer oxygen and CO2. Sickle cell anemia is a very interesting one. Hopefully we'll get to talk about this one a little bit more later on in the semesters. And it essentially means that the red blood cells are no longer donut shaped, but look quite frankly like a sickle. It's an adaptation to malaria that took place particularly in Africa, but it's led to a rise in the presence of sickle cell anemia as an inherited trait because of its ability to help an individual resist malarial infections. And finally, thalassemia. So this is also an additional adaptation to malaria, where irregular blood cells that contribute to lower abilities to carry oxygen have also been shown to reduce malarial infections in populations and increase survivorships in those areas. Polycythemia is very interesting and quite further on the other end of the spectrum. So it means we have too many red blood cells. The blood gets thick and heavy and sluggish in the veins. Other red blood cell problems can include things like cancer, leading into the red bone marrow itself, disrupting red blood cell regeneration. Also adaptation. A lot of us here at 5,000 feet have some altitude-based adaptations. This is not pathological, but can actually occur rapidly in just a few days. If you've ever gone running at sea level after living up in Albuquerque for a while, be ready to throw on those extra miles like it was nothing. It's kind of fun for that reason. However, the sense of danger also can increase and make the blood flow literally quite slow. It's impairing circulation, and it also might risk things like blood clots. So if it makes your blood run cold or run slow, that's quite a literal term. Another one is a little interesting, it's kind of scary, this idea of blood doping. So this is done by athletes who actually take out their red blood cells, like withdraw them, and then replace them later on right before they compete. And it means that the oxygen capacity goes up for their blood. However, it does mean their blood gets very, very thick. So that's in the polycythemia category. It's no secret that men and women have different levels of anemia, and that's usually pretty specific to presence of iron. So men tend to be able to carry around about 3.5 grams of iron. That's for, a few reasons. This is an average population where men are physically larger than women, so they physically can make more. And women also are physically smaller in many categories, so they typically have less. However, women also go through periods of, quite frankly, periods, right? So there's blood loss, childbirth. Oftentimes some individuals elect to not put supplements into their diet, or are as cautious as maybe they need to be when they're undergoing those biological processes, that they don't replace their iron reserves, and so that kind of has a cascading effect. All right, so this was the erythrocyte, an introduction to red blood cells. I know we went through a lot, but if you have any questions, please feel free to email me. Hope you have a great day.